Good afternoon, one and all. Today topic is Bentham and Hooker classification. This is important for essay 1. George Bentham and Joseph Dalton, the two English taxonomists who are closely associated with Royal Botanical Garden, England. They gave outstanding system of classification phenerograms in their book Genera Plantarum, which was published in 3 volumes between 1862 to 1883. Already told you yesterday, it is a type of natural system of classification. There are 3 types of classification: artificial, natural system of classification, and a phylogenetic one. So Bentham Hooker's classification, one of the natural system of classification. In natural system of classification, they have taken all type of possibility external characters. So this they described 97,205 species flowering plants, 202 orders. So in their classification, if you see their classification, plant kingdom, uh, not here, just one. Right. In his classification, actually, he only focused on phanerogam. But remember, in plant kingdom, actually, what happened here? Plant kingdom, phanerogams are there, cryptogams are there. These are the cryptogams. They are the non-flowering plants and flowering plants. But the Bentham Hooker focusing on only phanerograms, not cryptogams. What are cryptogams? Non-flowering plants such as algal member, bryophyta, pteridophyta. These are cryptogams, right? But it's not did not cater about these cryptogams. And phanerogams, they classified three classes, three classes. Class 1, dicotyledone. Class 2, Gymnosperme. And 3, monocotyledone. If the characters all inside the seed, if the two cotyledon present, they are grouped in dicotyledone class. And gymnosperms, seed present but not enclosed in fruit, openly formed. Seed present but it is not enclosed in fruit, but they are openly formed. The type of all grouped in gymnosperms. That means without fruit seed present. Monocotyledone. One cotyledon in seed. In seed, two cotyledon, that is dicotyledon. One cotyledon, that is in monocots. And again, the dicots class divided into 3 subclasses: polypetale, gamopetale, and monoclambed. What is characteristic feature of polypetale? Let's see. Poly means free here. Actually, the poly means free. Petal means corolla. And these petals are not fused. So they are all grouped in polypetale. And gamma petale. Next one, gamma petale. In gamma petale, gamma means fused. The petals are fused in gamma petale. Petals C fused. This is the symbol. Petal C not fused, open. Monoclambed. Monoclambed means in this calyx corolla, both form one type of structure that is called perianth. So this is grouped in monoclamidae. And the subclasses, these subclasses divided into series. These subclasses divided into series. For example, uh, Thalamiflorae, one of the series. Let's see full diagram. Thalamiflorae, which has 6 orders and 34 families. Disciflorae, 4 orders, 22 family. Calyciflorae, 5 orders, 27 families. So in gamma, first let me complete about series of Thalamiflorae. What are the characters in Thalamiflorae? Thalamiflorae, flowers with a flat, convex or rarely elongated thalamus, and which are hypogynous flower, and as it is polypetalous, sepals free, ovary superior, disc absent, divided into 6 orders. Divided into 6 orders, that is Thalamiflorae. So the flowers which are flat, convex, rarely elongated thalamus, those type of characteristic flowers are all grouped in Thalamiflorae. And again, Thalamiflorae divided into 6 orders. The 6 orders, order 1, if you have the pen and paper, write down here the 6 order, the first order, Ranales. First order Ranales, let me draw. Put a pen. Six order, the first one, here, Ranales. Second, Varietals. So time-based process, just write down. Ranales, and second one, Varietals. Third order, Polygonian. Fourth order, Caryophyllinae. And fifth order, Guttiferales. Sixth order, Malveals. So these are the 6 order. In examination, you just write the order, 6 orders. Thalamiflorae one series. You should write the series name. And also you should write the characters also. Thalamiflorae, disc shape, elongated, thalamus. That should write. If possible, write the 6 orders name, but not characters of the orders. For example, what are the grouped in Ranel's family? In Ranel's order. In Ranel's orders, if we take the flower stamens are free, carpels numerous, infinity, and these carpels are arranged in spirally. So this type of characters are grouped in Ranel's order, and this order again divided into 8 families. In in Ranel's 8 families divided. Actually, total Thalamiflorae 34 families. But in Ranel's have the 8 families. The families are Rananiculaceae, Delineaceae, Caliocanthaceae, Magnoliaceae, Anonaceae, Menispermaceae, Barbidaceae, Nymphaceae. And second order, Perietals. If you take the Perietals, the carpels are fused. Unilocular ovule, unilocular ovary, marginal placentation. These are the characteristic features of Perietals. If you take Ranel's, stamens free, but here carpels fused, carpels numerous. Here only unilocular carpel, unilocular oval, marginal placentation. And again it is divided into 9 families. Serraciniaceae, Papaveraceae, Cruciferae, Caparidaceae, Tresidaceae, Citinae, Violaceae, Cancelaceae, Bixineae. These are the different families. Next, order 3, Polygoniae, the compound gynoecium, Bilocular ovary. So these are the characters of polygonal, and it is divided into four families: Pictospermaceae, Tremendriae, Polygalae, Vachiaceae. So these are the four families. And fourth order, Caryophyllinae. Actually, the orders end with ales, but here order the Polygalae not end with ales, and Caryophyllinae this is also not end with ales. And in Caryophyllinae, the free central placentation, that is the important character of this feature. There are four families: Francaniaceae, Caryophyllaceae, Portulaceae, Tamaricinae. Here also not end with AC, EA. And fifth order Guttiferales. In Guttiferales, sepals imbricate distribution, one character, stamens numerous, another character, and it has the six families. Elatinae, not with AC here also. Hypercinae, Gettiferae, Ternstromiaceae, Diptocarpaceae, Claniaceae. So these are the six families. And the last order, sixth one, Malvaceae. In Malvaceae, sepals free, valvate distribution, stamens numerous, carpel number 3 to 8, fused, exile placentation. So this is the Malvaceae, and it has the three families: Malvaceae, Sterculaceae, and Tiliaceae. Second series is Disciflora. So first Thalamiflora completed, and second series in Polypetale, Disciflora. Disciflora name itself given disc, that means the thalamus disc-like, flat, slightly convex, and sepals free, adnate to ovary, hypogynous, disc present. Hypogynous. In Thalamiflora also hypogynous, and here also hypogynous. Andrecium definite, ovary superior. Maybe it is hypogynous and ovary superior, divided into four orders. In Disciflora, four orders. The orders are Geraniales, Polacales, Silastrales, Sapindales. Four. The next one, and let me complete end with the families also. In Geraniales, there are 11 families: Lineae, Hemriaceae, Zygophyllaceae, Geraniaceae, Rutaceae, and Semerbiaceae, Cochineae, Brucerceae, Miliaceae, Chiloideaceae. These are the families present in Geraniales. And Olacales, second order, only three families: Olicinae, Illicinae, Cyriliaceae. And third order, Silatrales. It has the four families. And fourth order, Sapindales, three families: Sapindaceae, Sabiaceae, Anacardiaceae. So these are all grouped in Disciflora. And another series, Calasiflora. And next series, Calasiflora. In Polypetale, three series: Thalamiflora series, Disciflora, Calasiflora. In Calasiflora, thalamus concave, sepals are fused and adnate to gynoecium. Flowers perigynous and epigynous. Perigynous means it is partially not hypo and not epi. So that is called perigynous. And few of the families are epigynous, and it is divided into five orders. The order one is Rosales, which family again classified into eight families. And order two Midtails. Midtails, it is also the classified into seven families. Order three Polyflorales. It is also divided into seven families. Order four Ficodales. Order five Umbellales. So the five orders in Calyphlora. And second subclass, that is Gamma Petale. Already told you Gamma Petale character. The gamma-petalic characters biseriath perianth, like a polypetal biserianth means sepals petal separate. Here also sepals petal separate, and petals are fused. Here the petals free. Petals are fused. And epipetalous condition. Epipetalous condition means calyx, corolla, andrecium present. Epipetals means if andrecium fused with corolla, that condition called as epipetalous condition. In this have the inferae series, three series here also, inferae, heteromerae, bicarpellate. First, let's see the inferae family. In inferae family, all flowers are hypogynous. Stamens as many petals are adnate with them. Ovary inferior. Simple. Inferae inferior family. Divided into three orders. The first order is rubiales. In inferae, first order is rubiales. This rubiales, the containing two families, Rubiaceae and Capriliaceae. Order to Australes. It is important order Australes. In Australes order, which is divided into four families, example, Velernae, Compositae or Austraceae, Dipazaceae, Calicerae. These are the families. Order 3, Campanules. And Campanules have the three families. And second series, Heteromerae. Second series, Heteromerae. In Heteromerae also have the three orders. Heteromerae, three orders. The three orders are Ericales. It has six families. And Primules, three families. Ebineles, three family, three. So total inferae, nine families. Inferae, nine families, and each series, three families. Sorry. Heteromerae, sorry, how many families? Heteromerae, how many? Heteromerae, here where it is. Right, Heteromerae, three families. Sorry, Heteromerae, total families 12, total orders are three. In first order, that is Ericales, in first order, six families. Second order Primules, three families, 6, 3, 9. And Ebineles also three families. Total 12 families in this. And next third series, bicarpellate. The name itself given bicarpels. So the number of carpels two. The flowers hypogynous, carpels two fused, ovary superior, and it is divided into four orders. The four orders and 23 family. Four orders and 23 family. The four order, the first order is Gymnosperms, which has six family. Gymnosperms, which has six family. Order 2, Polymonials, it has five families. Order 3, Personales, it has eight families. Order 4, Lamiales. Lamiales again four families. Total 23. Total 23 families. And another subclass Monoclamidae. Now Polypetalae completed, Polypetalae series completed, and Gamma series and orders completed. Now the Monoclamidae. Monoclamidae uniserianth, perianth. In Polypetalae and Gammapetalae bi. But in Monoclamidae, uniserianth perianth or perianth reduced. If it is perianth reduced, that means the petals absent. Maybe petals absent. It is divided into seven series. is divided into monoclamidae, divided into, where is the monoclamidae, divided into seven series, and the series have not have further divided into order. There is no order, only the seven series are there. Series is seven series. Curvambriae, Curvambriae, direct six families. And second, multi, sorry, multiovaliate, divided into one family. Multivalent terrestris, aquaticae, aquatic and terrestris. Aquatic one family, terrestris three family. Microembrya four families. Microembrya four families. Next, Daphnails, another series. Aclamdospore, three families. No orders. Unisexuals, nine families. Ordinaceae, nine families. So total seven. Curvambria one, multivalent aquatica two, multivalent terrestris three, microembrya four, one, two, three, four, Daphnails five, Aclamdospore six, unisexuals seven. Seven orders. They are directly into families. Directly into families. Next. So this is all about only dicotyledons and dicotyledon subclasses completed. Now, next class, gymnosperms. Already told you gymnosperms, presence of naked seeds is an important feature. There are three families. Only gymnosperms, they know subclasses, no series and orders directly divided into three families. The three families are Neetaceae, Neetaceae, remember, Neetaceae, Coniferaceae, Coniferaceae, Cycadaceae. Neetaceae, Coniferaceae, and Cycadaceae. These are the three families. And class 3 is monocotyledons. Monocotyledons, already told you, one cotyledon, that is the main character feature and advantageous root system present. Parallel venation flower, sorry, parallel venation in leaf, and it is divided into seven series. This is also like a monoclamidae, which also divided into seven series. And the seven series are Microspermae, which has three family, Ediginae seven family, Coronariae eight families, Cyclocinae five family, Nudifloro five family, Apocareae three family, Glumaceae five family. These are the monocotyledons, right? Monocotyledons also no orders. 1 minute. There is no orders, right? So this is Bentham-Hooker classification. In examination point of view, just draw this flow chart and write merits and demerits. Let's see the merits and demerits of this Bentham-Hooker classification. Right. So the main features already discussed. Let's see the Bentham merits of Bentham and Hooker system of classification. Uh, First merits. Let's see the merits. The description of families and genera is very accurate. Family description, description, very accurate. The system is very handy for identification purpose, and the system is of great practical convenience. The British and Commonwealth Herbaria, therefore, they still adopt this system in arranged families because the most of the family identification and classification done by Bentham-Hooker classification. Now the most of the countries follow this type of classification. That's why it is the great practical convenience the British and Commonwealth Herbaria. Each family had a synopsis at the beginning, which are very useful for identification of the plant. The system start from Ranel's order, which are now universally considered to be the most primitive living angiosperms. That is also one of the best merit. Larger genera subdivided into subgenera and sections for our convenience. They believed in evolution through the reduction and hence placed the monocot after dicots. According to them, the evolution complex to simple. That's why through the less parts and considered as advanced one. That's why they believed that evolution through the reduction and hence placed the monocot after dicots. First dicots, later monocot. Even in dicots, dichlamydases, polypetale dichlamydase means perianth. There is no perianth. Two sepals and petals are there. That is the first free, the fused. Later uniperianth, the uniseriate monoclamide. And the gamma petale placed after polypetale, that is also justification. The polypetale includes thalamiflorae, the first, calyciflorae, that is also follow the evolution process. But the Bentham Hooker distinguished new series Disciflorae, which includes orders which cannot be assigned to Thalamiflorae and Calyciflorae also. The three series Thalamiflorae, Disciflorae, and Calyciflorae show gradual evolution, advance from the marked hypogamy to epigamy. First hypogamy primitive, now perigamy, epigamy. Treating Cucurbitaceae and Umbelliferae at the end of the polypetale as connected links between poly and gamma petalous families. Right, because actually this Cucurbitaceae, Umbelliferae, Apiaceae families, some of the plants, actually they are in polypetals. Actually in polypetals, the petals are free, but instead of that, somewhere the connection there, fused. That's why this is the link between the poly and the gamma petale. And now creation of monoclame at end of the dicots, that is also good decision. Merits of Bentham-Hooker system next to disputed families included ordinals, anemone, and placing unisexual monocot families after the bisexual families. That is bisexual means complex, two parts, unisexual one part, that's simple. The series Glumiaceae with extremely reduced flower and inflorescence placed end of the flowering. So Glumiaceae flower parts reduced, completely reduced. Now the system was never conceived by its author at the base of phylogeny. The theory organic evolution theory was renounced independently by Darwin in 1859. Only the criticism the system on the base of phylogeny it is not too justified. And some of the other merits. So the classification is not a phylogenetic one, but some of the character shows the phylogeny. The Ranelian flowers with an elongated floral axis, spiral arrangement part, that is the primitive character. The system of classification has a practical value and easy workability. So these are the most merits. Let's see the demerits. Let's see the demerits. The system does not give any idea as to the evolution history of any genus or family order. Actually, it did not cater the evolution process, but nowhere their evolution process are there, but not follow the completely. In this system, the grouping of plants is mainly based on single and artificial character, which is not good enough for classification. The single character, artificial character, what is artificial nomenclature classification? Artificial classification, one or two characters take consideration. With the result that close allied families are placed widely apart, and the group of monoclamidae entirely artificial. Monoclamidae only one character. That one character is perianth, or maybe the reduced petal. That's it. That is also not proper way. And another, the blunder one, gymnosperms is placed between the dicots and monocots. If you take the evolution process, algal member, bryophyta, pteridophyta, gymnosperm, angiosperm. But where is gymnosperm in between angiosperm? It is not a correct way. So this is also another the biggest mistake he has done. They have done. And the system does not show the phylogenetic relationship already told you because it is not a phylogenetic first. The main demerit is that the system does not give us any idea as to evolutionary history. The one family or other family, how it is evaluated, how it is primitive family, how it is advanced one. Unless the one character, the complex one, primitive, simple one, advanced one. And composite Asteraceae, actually it is highly advanced family and which is placed in inferae in the beginning of gamma petale. Actually, whether it is phylogenetic classification or not, but to follow the some of the character phylogenetic. The advanced must be in last, but he placed in the first in inferae. And another advanced families like Archidaceae and Skimnaceae are treated as beginning the monocot. This is also another blunder mistake they have done. And Liliaceae, Amaryllidaceae were kept apart through they are very close related. Liliaceae and Amaryllidaceae were kept apart one single branch because they both are close related. And the position of series Apocarpe is unsatisfactory due to the free and superior carpels. So these are the 10 demerits. And right. Let's see the angular plantal classification. This is also important one. Angular plantal system of classification. Angular plantal system of classification. Let's see. 1 minute. Angular plantal genetic. Uh, Let me take PPT slide. Physiology. Secondary growth. Where is angular plantal? Anthals, Mr. Anthal. Also. Angular plantal. Bentham hooker. Okay. Just wait. Angular plantar algae. Bentham hooker. Cell Clamdomonas cell nature. Epigenetics, incidental character. Notice about gene expression, nomenclature, physiology, plant gene, prokaryote translation. The ultra-structure typical vector cell transcription eukaryote, Archaea alba. Angular plantar somewhere I think it is principles of priority. Okay. Angular. Okay. Angular and plantar system of classification. The Adolf Engler and Carl Plantal. 1 minute. Adolf Engler and Carl Plantal in 1887, 1899 presented a phylogenetic system of classification of their 23-volume work Dinaturalism Flangen Familien, where in all the nose genera from algae to angiosperms they have been classified. But in what done in Bentham and Dokar only phanerograms did not cater the cryptograms, but they classified from the algae to angiosperms. The system has been revised by Gilg scientist in 1936 and underwent several changes. In the system, the gymnosperms are separated from the precede angiosperms. And what happened in Bentham Hooker classification? In Bentham and Hooker classification, actually, in angiosperms, between the angiosperms placed, and he just before the angiosperm placed the gymnosperms. That means gymnosperms next angiosperm. That is, it follow the angiosperm evolution process. And it is framed on the presumption that evolution that proceeded from the simplicity to complexity flora. Bentham Hooker complex to simple, but he has done simple to complex, reverse manner, right? Let's take 2 system. Okay. Right. So these are the both are the angular and plantal. So they published the book, that is the 23 volume monumental work, De Natural Plants Familion. They classified algae to angiosperms, and the system has been revised. Gilg scientist one time, and later Ernst Mayklair published the revised edition of the syllabus in 1964. So their concept, let's see their concept. Already told you, the first of all, the gymnosperm placed before the angiosperm, that is the most important character. And another one is simple to complex. Among the dicotyledon, there is a amalgamation of eclambidae and monoclambidae taxa with this polypetalous one into the one group, namely Archiclambidae. All the sympetale taxa among the dicot grouped in under the one branch, that is Metaclambda sympetale. Monocotyledons are placed prior to dicotyledon. That is, I think this one not to follow the phylogenetic. We will discuss later merits and demerits. And the unisexual naked flowers arranged in catechin and wind pollinated were considered in the lowest grade of flower organization by Engler, and as such is called Englerian concept. The next stage of evolution in flower followed by the appearance of one seriate perianth leading to two seriate condition. One seriant, it is simple. Two seriate, advanced, complex. Apetalous, first apetalous, petals, no petals. Polypetale, petals free. Sympetale, petal fused. First actinomorphic, to zygomorphic. Unisexual to bisexual. Hypogynous to epigynous. These all character reverse in Bentham and Hooker classification. Simply, it is opposite to the Bentham and Hooker classification. And placed the gymnosperm first. That's why they discuss about gymnosperms. The subdivision gymnosperms has been definition same. What the given definition Bentham and Hooker, the same subdivision gymnosperms has been divided into 7 orders. The starting one being Psychedophyllales, the most primitive one, whereas the ending order is Nettles. Psychedophyllales. But what happened in the Bentham and Hooker classification? First Nettles, last Psychedales. But in his order, Psycheds first, Nettles last. And it is advanced, of course, it is the advanced character in gymnosperms. Most of the Nettles plant shows the angiospermic characters. The plant kingdom has been divided in 13 division. Division 1 to 12 dealing with bacteria, different type of algae, fungi, bryophyta, pteridophyta, all. So that is the first 1 to 2 division. And 13th division named as a embryophyta and Siphonogamia, which includes all the seed producing plants. So from pteridophyta 1 to 12, all non-seed plants like cryptograms. But in 13th division, last division, only phanerograms, but the named as embryophyta, which has contains embryophyta Siphonogamia, which includes the all seeded producing plants, which is divided into two subdivisions. Gymnospermae first, naked oval seed, same character, where the told by the Bentham Hooker. Angiosperm enclosed ovules in seeds. Gymnosperms has been divided into 7 orders from Psychedophyllales to Nettles. The subdivision Angiosperms divided into 2 classes. Angiosperm first monocotyledone, second dicotyledone. Next monocots. The monocots divided into 11 orders and 45 families. First order is Spontaneals, which is possess the naked unisexual flowers. That is simple. The last order Microspermae with Orchidaceae, the last family. Microspermae, the complex flowers are there. and dicot character. Dicots are divided into two subfamily. Already told you Archiclamidae and Sympetale. In Archiclamidae, the flowers are eclamitous or diclamitous, but petals mostly free, polypetalous condition. So in Archiclamidae, the monoclamidae are diclamidae, both one grouped in one Archiclamidae, but in this only apart from the monoclamidae, in diclamidae, polypetalous condition present. In Sympetale, petals are fused. Eclamidae, second Sympetale. Outline basis classification. Archiclamidae has 33 orders and 206 families. The first order being Verticillate and Casuarinaceae. The last order being Umbelliferae with the Carnaceae class family. Sympetale, 11 orders. Sympetale or Metaclamidae. Sympetale or Metaclamidae, remember. 11 orders with 52 families. The 11 orders and 64 families revision Hans Meckler 1964. So extra some families are added. The first order being Ericales with the Clithiaceae family. The last order Campanuales, Ostraceae. This is also follow the phylogenetic. And the plants first to layer. This is important. In examination point of view, just draw this table and explain to everyone. Plants divisions, 13 divisions actually, 1 to 12 bacteria, pteridophytes. Actually, bacteria, algae, bryophyte, pteridophytes. You should write all. Should write back or simply write as a cryptograms. Bacteria, algae, bryophyta, pteridophyte. That is very good. And 13th division, embryophyta, siphnogamia. In embryophyta, siphnogamia, gymnosperms and angiosperms. First gymnosperm, next angiosperms. In angiosperm, first monocotyledon, next dicotyledon. In dicotyledon, Archiclamidae and Metaclamidae or Sympetale. Right. So this is classification. Let's see the merits. If we see the merits, the system deals with the all group of plants. That is the best merit from the bacteria to the angiosperms. It is a phylogenetic system. The plant groups arranged mostly according to evolution affinity. Primitive group followed by advanced one. And already told you, Gymnosperms treated as separate group in position before the angiosperms. And Asteraceae create a special place. And the common position of Orchidaceae also well positioned at the end of the monocots. And this is very accurate. It is the most evolved family monocots. Anatomical data were taken into consideration in this system of classification for the first time. So for an artificial and natural system of classification, mainly the taking consideration only morphological character, but not anatomical character, not evolution process. But here anatomical character also taking consideration. Monoclamidae has been completely abolished. There is no monoclamidae. The monoclamidae present in Archiclamidae. And its families have been distributed among the related forms with free petals in the large series of the angular systems. And it is intended to express the phylogenetic relationship already told you. Right, Composite and Archidaceae both are the given the best position. Right, demerits. Take demerits. Monocots, this is already told you. The monocots treated as a primitive. That is the blunder mistake they have done. It is not a correct according to modern evolution evidence. The concept of primitive flower, unisexual acclimatus is against the modern concept. This is also not follow them. Amalgamation Hepatalis family with Polypetalus family from the Archiclamidae is not a desirable, not a justification. The system is not much particularly utility. That's why the most of the followers follow the Bentham and Hooker classification. And derivation of the free central placental from parental placentation. Derivation of the bisexual flower from the unisexual flower. Derivation of the entomophily from the anemophily. These are all demerits. From parental placentation, free placental formation, and unisexual to bisexual, and then anemophily to entomophily. They are not a clear justification. Right. Draw this only this table enough in examination and explain one by one. Write merits and demerits. Next one. So what are the differences between the angler and planters? Angler planter differences. Take the differences between angler and planter. First, come to the author wise. Bentham and Hooker's scientist, angler and planter scientist. The Bentham and Hooker classified their notes in published in a book that is Genera Plantarum. The regular planter book is Dinature Plantar Familiam, which is 23 volume book, Genera Plantella 3 volume book. Genera Plantarum published 1862, 1883, which is 1887, 1899. This classification basic based on Jesu classification, Bentham Hooker classification based on Jesu classification and APD control. And based on Heikler classification, Engler and Plantel based on Heikler classification. And this Bentham and Hooker classification, uh, natural system of classification. And Engler and Plantel classification, one of the phylogenetical classification. Oh, and Bentham and classification only limited to phenerograms. In Engler and Plantar, the entire plant kingdom taken into consideration. Position of Gymnosperm between the dicots and monocots, but in Engler and Plantar placed before the Angiosperms. Evolution Angiosperms is not discussed, but here evolution Angiosperm discussed polyphyletic. According to Bentham and Hooker, the complex flower advanced, then simple flower, but the same character reversed in Engler and Plantar. Simple primitive, complex advanced. Position of dicot precede the monocots. Dicots first, later monocot. But here monocot first, later dicot. And the main tags are divided into polypetale, gamma petale, and monoclamide. But here, eclamide, sorry, here the polypetale, monoclamide, both are eclamidas taxa, and rest of the fusion gametes, fusion petals are in sympetale taxa. The main taxa monocots divided into 7 series, okay, divided into 11 orders. In Engler and Flanter, 11 orders. And Bentham and Hooker classification placement start with the ranels and Epocarpus flowers such as Ranunculaceae, Magnoliaceae, and end with Lamiales. And Engler and Plantal classification start with Castoriaceae, Faberaceae like family, end with the Campanulaceae that show flowers with biseriate perianth. The monocot start with the taxa Orchidaceae, ends with Glumiae. But according to Engler and Plantal, start with Pandales that show imperfect perianth and ends with Orchidaceae. The number of families in Bentham and Hooker 165 dicotyledons, 34 monocots total, 199. There are in angular plantal there are 239 dicotyledons and 45 monocotyledons. Total 284 families. Total 280 families are there. So these are the classification differences between Bentham and Hooker system of classification. And Bentham and Hooker system of class completed. Angular plantal completed. The both the merits and demerits also completed. And differences also completed. These two very important in exam point of view. And the next one, the not important, but we will discuss here. Taxonomy Hutchinson concept. First, let's see the Hutchinson system of classification and merits and demerits. The John Hutchinson from England proposed a system of classification. This classification also based on the Bessie. Bentham and Hooker classification D. Jesu. Engler and Planter classification Eichler. Hutchinson classification based on Bessie. He classified mainly the flowering plants. Only flower, like a Bentham and Hooker, he also classified only flowering plants. His phylogenetic system first appeared in two-volume work, the book named The Families of Flowering Plant. Families of Flowering Plants. And it is underwent several revisions also. The final revised edition appeared in 1973. Hutchinson accepted Bentham and Hooker concept of primitive flower. So almost Hutchinson concept is similar to the Bentham and Hooker classification. However, he abolished the monoclamidae and only two groups are recognized in dicot, namely Archiclamidae, Metaclamidae. The mixing the both angular and plantar, but mostly taking consideration Bentham and Hooker. But only has done one change. Bentham and Hooker, there is a three clear. One is gamma petale subclass, polypetale subclass, and monoclamidae. But here, only two groups recognized in dicot, namely Archiclamidae, Metaclamidae, like angular and plantal. The former were divided 59 orders and 213 families, while the later included 23 orders and 41 families. They are extra. He gives great importance to habitat plants. He considered that the dicot evolved along two lines of one of which predominantly two lines of evolution only. And the monocotyledons were divided into three distinct groups on the basis of nature of perianth. And these are the classification. These are the characteristic of natures of these Hutchinson classification. So this is also one of the phylogenetic classification Hutchinson. Let's see the some of the merits and the demerits of the Hutchinson. The system is based on considerable knowledge of phylogeny. The system based on phylogeny, considerable level of phylogeny. And it's based on phylogeny principle, okay. The treatment of magnoliales as the starting point of evolutionary series in dicotyledons in arranged with the prevalent views. Classification of monocotyledons is well received by contemporary authors. That is also one merit. The derivation of monocotyledon from dicotyledons widely accepted. The derivation of monocotyledons from dicotyledons. So actually, what the mistake did in angular and plantal, he has corrected. Let's see demerits. The series of objection has been primary division into woody versus herbaceous evolutionary line, basis of habitat. Is done woody plants were separate and herbaceous plants separated. So there are the most of the objections come forward. This leads to separation of some obviously close related families, Umbelliferae, Aracheraceae, Labiati, Verberaceae. Actually, they are the closely related family, but they are separated based on this character of wood and herbal. Hutchinson derives of angiosperm from proangiosperm. Of course, it's given, but it does not produce information about the nature of this hypothetical ancestral group. How the angiosperms derived from the proangiosperms, but did not give proper justification. He considered the Magnoliaceae the most primitive family, but most of the contemporary authors considered the Vezellus Winteraceae to be the most primitive. Right. So Hutchinson classification tables given page number 181, but no need to write all. Just give a table, and this in this table you just write number of divisions, orders. That's it. No need to write all families. And another one, Taktajan. Very good. Taktajan given or not? Next, Taktajan classification. Harman L. Taktajan proposed phylogenetic classification Nair plants and the actual system of classification based on phylogenetic principles. He was inspired by the Hellier, who created synthetic evolution classification flowering plants based on Darwin philosophy. Based on Darwin philosophy, Taktajan considered the order Magnoliaceae sissulatae as the most primitive, like Hutchinson. He derived monocotyledons and nymphaeals from the hypothetical common ancestor with vesselless wood and mononepetal pollen. He revised the in the revised classification published in 1966. The most important revised appeared in 1980 Botanical Review. In this revision, he recognized two classes, Magnoliphyta, consisting seven subclasses, 20 superorders, 71 orders, and the triple three families. And also, he also done only flowering, classified the flowering plants, but not all get to angiosperms. Here given page number 191, some of the important subclasses and order. But you just write the subclass and order, but not you did not write the family name. Just write the number. For example, order subclass Magnolidae. If the class is Magnoliopsida or Dicotyledon. The subclass is Magnolidae. Order is Cyclanthales. In Cyclanthales, one several orders are there. Later in each orders, the family is different family. Magnolias, 8 family, Iliaceae, 2 family, like that. Just name the number. 2 families, 3 families, number enough. Right? Division name, subdivision name, order name, just number of families. And merits, let's see the merits and demerits of this Taktajan. So let's see the first merits. Taktajan has taken into consideration all available evidences such as morphological, anatomical, embryological, palynological, cytological, ultrastructural into consideration while the classifying the plants. So morphological and anatomical, not only morphological and anatomical, only the angular plantal anatomical morphological character taken, but embryological, palynological, cytological, ultrastructural consideration, all this. The system is accordance with the rules and regulation of International Code of the Botanical Name Nomenclature. So this is the ICBN. It strictly follow the ICBN rules, and it suit for ICBN rules. And the demerits, the main objection to Takthajan system is derivation of monocotyledons from the stock ancestral the nymphaeads. He could not give proper justification about this. Concrete classification not important. So these are the different type of system of classification, right? And next one, recent trends in plant taxonomy, anatomy, palynology, embryology, cytology, chemotaxonomy, numerical taxonomy, and molecular taxonomy. These are all. First, let's see the anatomical relations. In anatomical relations, in taxonomy, taking for identification of plants, take the consideration epidermal characters and trichomes, epidermal cells, petiole anatomy, food anatomy, floral anatomy, fruit anatomy. They are all. First, in anatomic character, first let's see the epidermal characters. In epidermal character, first one stomata. Stomata based they are different type of stomatals: anomocytic, anocytic, parasitic, diacytic, tetracytic, actilocytic, cyclocytic type, exocytic type. Already they are discussed in anatomy class also. In this anatomy class, these are all type of stomatal type we discussed, and also you must have learnt in practical training classes also. No need to explain here. Don't waste your time and my time. After that, and next one is trichomes. The trichomes also another important anatomical feature character. Based on trichomes also, we can classify the plants. These trichomes are epidermal outgrowth. The classified them posed on considerable problems. Trichomes are great systematic significance and often common types are used for diagnostic purpose in association with other characters. The recent trichomes classified the recent trichome classification is devised by Theobald et al. 1979. The stars recorded that the trichome's anatomy is of immense significance in the classification in the family Combretaceae. The most recent classification, Theobald et al., that is practical and straightforward. And next, another important character based on epidermal cells also. The epidermal cells can provide the host features, and example their use abundantly occurs in literature. These include their shape of the epidermal cells, that is the different to one species to another species. Size also different, orientation difference, anticlinal and wall undulation, and periclinal wall cultures, curvatures. So the epidermal cells modification occurs also on the leaf margin, and when it is silicified, calcified, suberized. And some of the epidermal cell consisting different type of chemical, that is calcium oxalate crystals and foliar crystals found to be useful classification. Based on this calcium oxalate and also foliar crystals that can classify the plants is useful for taxonomic aid. And succulents and arid plants which belongs to arid region possess latispherous cells. So in epidermal cells it consists in latispherous also. They vary widely their structure and latex in their composition. And next one, another anatomic character, petiole anatomy. Stomata anatomy completed. Second one, trichomes. Third one, epidermal cells. And petiole anatomy. To take the petiole character, the vascular systems, distal end of petiole dicotyledons of considerable taxonomic importance. The vascular structure of petiole is much diagnostic significance in the genera Chlomis and Aristarchus of Labiate. So in petiole, there are two distinct vascular bundles arranged. The first one to two median arcs in noted it is noted in Flemish section, which is species of the Yr-MSc. Second type is characters occurrence numerous separated bundles, not only two, number of separated bundles, complete cylindrical occurs in Chlomis. In herbaceous number, the stem anatomy has proved to be the sum of diagnostic valve. During the last 5 decades, the plant's anatomy, notably mainly its associations, have established a number of lines of evolutionary specialization in true structure of steel. Like, yeah, actually, the Bentham and Hooker split the Genetiaceae into two families, Miniathenidae and Genetiaceae, based on this character. Vessels are evolved by the modification of tracheids. The most primitive vessels have members closely resemble tracheids. Tracheids cannot be derived from the vessels. And wood anatomy relations to taxonomy. In wood anatomy, if you take some of the family Winteraceae, first of Winteraceae and Umbrellaceae, Tetrastaceae. In this group, the actually wood is actually based on the data from the wood anatomy are considerable taxonomic and phylogenetic significance. At generic and below the generic level, the most useful contribution of wood anatomy is to support the placing taxa uncertain affinity. With the help of anatomical evidence, Fabaceae different subgenera and section can be delimited with evidence of wood structure. The placement of the genus Myristica and related genera close to Laraceae. Similarly, based on wood structure, true evidence placing Calecanthaceae and near to Rosales, Myrtles, and Pteridophyta Gymnosperms along with Angiosperms, Astrobolia scandes exhibited unspecialized sieve cells in phloem. Floral anatomy and taxonomy. The affinities of families based on the floral anatomical patterns in Papaver, Capparaceae, Brassicaceae, and Magnoliaceae could be established. The families commonly exhibit partial placentation and inverted placental bundle with regard the floral axis, inner side of secondary marginal stand with normal orientation. This fact also suggests the placement of Moragineaceae and radials. But the scientists concluded that the Cyctadinae has no close affinity with members of Genseriaceae and suggested it transfer the Scarpiaceae with it exhibit more resemblance have supported the treatment based on the floral anatomical character. And another anatomy character, fruit walls and seed coat anatomy. So these are all anatomical characters. Next, embryology relation to taxonomy. So embryology feature mainly take the consideration anther, pollen grain, ovule, archisporium, megasporogenesis, which is development to embryo sac, mature embryo sac, fertilization, endosperm, embryo, seed coat, and special features. So first of all, let's see in the anther, the maybe the number of vertical rows taking the consideration, the wall development basis, the wall development also that is monocotyledon different, dicotyledon different, tapetum, in tapetum glandular, amoeboid tapetum, number of ploid nuclear lateral cells, successive assembled cytokinesis type, they are the taking consideration character to classify the plants. Pollen grain, an NPC system based on pollen grain NPC number, position and character. So the one plant to another plant, the number of pores different, the pore position different, aperture types also different. And another one is in types of ovule, orthotropous, anatropous. Already we have discussed this all type of ovules in anatomy and also in practical examination also. Orthotropous, anatropous, compylotropous, hampitropous, circinotropous. So they are all, these also taking consideration one of the characters, embryological character. And archaeosporium, the number of archaeosporium cells extends to parietal tissue. The sum of the species different to another. Megasporogenesis development. After megasporogenesis, that means archaeosporial cell developed into megaspore mother cell. This megaspore mother cell divide mitotically and form megaspore. This megaspores later developed into embryo sac. This embryo sac may be mono, bi, tetra. Mono means only one megaspore developed into embryo sac, that is the monosporic development. Bisporic means two megaspores developed into embryo sac, that is bisporic. And tetrasporic means four megaspore developed into tetraspores. Next, mature embryo sac. Number and organization of cells in the embryo sac, that mean so any development, whether it is monosporic or bisporic or tetrasporic, after formation embryo sac, the embryo sac also mainly have the eight nuclei, seven cells. So this eight nuclei, seven cells, the seven cells are the first filiform apparatus, three cells, egg cell, two synergids, central secondary nucleus, one cell with two nuclei, and lastly antipodal cells, three cells, three nuclei. So this arrangement different after maturity. This arrangement different in mature embryo cell and based on fertilization also. Path of entry of pollen tube. Maybe some family porogamy, some family shows mesogamy, some families show chalazogamy. That means the pollen tube enter into the embryo sac through the micropore, that is porogamy, or through the integument, that is the mesogamy, through the chalaza side opposite to the pore, chalazogamy. That is also one of the characteristic features to taxonomic aid. And embryo, so endospherm. After fertilization, embryo and endosperm both are formed. That is called bifertilization. But in this first maturity nuclear, endosperm. That's why let's discuss about endosperm. And endosperm also three types based on the species: nuclear type, cellular type, heliobiyal type. And also not only this different type and this different type with or without ostoria. Some of the endosperm with ostoria and few are without ostoria. And also the seeds, albuminous seeds and exalbuminous. That means endospermous, non-endospermic seeds also there. They are also, they're all one type of character, taxonomic aid to classify the different plants. Seed coat, embryo, sorry, next embryo. Development type, asteroid type different, anagrated different, kinopodial different, solenoid different, paraphyllate different. Already In embryo, actually, is not in the C3. So, but anyway, remember this development of asteroid, onogroid, phenopodial type. That embryo also not unique development, different development. So, this is all different type of separated into taxonomic group. Seed coat, number of layers and their derivations from one to one integument. And special features apomixis and polyembryony. What is apomixis? Apomixis means that without fusion of gametes, directly without gamete fusions, directly the seed formed, that is called apomixis. Polyembryony, that is one of the another character. Polyembryony means actually after fertilization, one embryo formed. Instead of that, more than one embryo formed inside the embryo sac, that is called polyembryony. The polyembryony, true polyembryony, and false polyembryony, that is the based on different species. So this is embryonic study, and later some given um, phylogeny. Next, we will discuss about phylogeny characters and molecular level taxonomy, chemotaxonomy, molecular taxonomy. Next class, we will discuss all this. Thank you all.